I have the great pleasure to interview today Haitham Amal. He is head of the Laboratory of Neuromics, Cell Signaling, and Translational Medicine website, visiting professor at Boston Children's Hospital, Harvard Medical School, and associate professor at the School of Pharmacy, Faculty of Medicine, Hebrew University of Jerusalem. He wrote 2 amazing articles, and his laboratory right now is working 70% of his time, more or less, on nitric oxide and its application for autism. So welcome, Haitham, and tell us more about nitric oxide. Many people have no idea how important it is.
Nice to meet you, and thanks for the invitation. Nitric oxide is a small molecule that is produced in our cells. Actually, the one who discovered this was Professor Steven Tenenbaum, who was my mentor at MIT. So nitric oxide produced in our cells and plays a critical role in the body. But today we're going to talk about the brain, and nitric oxide acts as a neurotransmitter that affects neuronal migration, synaptic plasticity, neuronal function, behavior, and many other cognitive functions in the brain.
A Nobel Prize was attributed of physiology and medicine for discovery of nitric oxide. And if we know something about it, we know the nitric oxide secreted by the endothelium, and that creates vasodilation, work on the adhesion of the platelets. And so I remember there's 3 isoforms of nitric oxide. And the one we want to discuss today is the neuronal nitric oxide, not the endothelial, or the other one is inducible.
Right.
Right.
Exactly.
So the neuronal, my question is, also create a hypokalemia, hypotension, vasodilation, or it doesn't have these function at all?
Yeah, so we believe that the neuronal nitric oxide synthase is very specific to the brain and neuronal cells. It also located in peripheral neurons, but most of the effect for vasodilation and heart hypertension is related to the endothelial nitric oxide synthase. And therefore, in all of our applications to treat autism and Alzheimer's and different brain cancers, is that we are focusing on neuronal nitric oxide synthase that is overactivated, that leads to overproduction of nitric oxide and then to the pathology.
Yes, so autistic child overproduce nitric oxide, but that doesn't give them vasodilation, thrombocytopenia, low blood platelets count. Sometimes they have, hyperbilirurimia. So all those doesn't happen in autism. It's a totally different effect in the brain.
Yes, we still didn't test nitric oxide in the brains of humans. We did in mouse models and with human induced stem cells, as well as blood sample of autistic kids. We believe that there's an overproduction of NO in the brain that actually, of course, doesn't affect the heart and blood tension system.
You found out that the autistic child have overproduction of nitric oxide. Do you find out why it stimulates this neural nitric oxide synthase? Why do they have more nitric oxide when autistic?
Yeah, so as I said, the story started when I was at MIT as a postdoc there. And what we figured out is that there are mutations that leads to autism, such as SHANK3, leads to increase of calcium influx into the neurons. And the calcium that influxed into the neurons lead to overactivation of the neuronal nitric oxide synthase. So this enzyme, which is a nNOS, is a calcium-dependent enzyme that is activated by calcium. And this is the mechanism that we think that leads to overproduction of NO. It's a calcium-dependent manner.
But I remember the neuronal as well as the endothelial nitric oxide are both calcium dependent, not the third type, not the inducible nitric oxide. Because of this calcium, this gene that modifies the calcium, we don't have any action on the endothelial nitric oxide in autism.
If we talk on the brain, we think that the only enzyme that is affected is the neuronal nitric oxide synthase. Generally speaking, we did some toxicological studies and we didn't see any change in the hypertension, any change in motor activity, any change in weight loss. And therefore, we think that the molecule that we develop is very specific to nNOS.
Yeah, so SHANK3, if I remember, is one of the most prominent gene we find in autism, maybe 1 to 2%. And
Right.
it's on chromosome 22, right?
Right.
And so you work also on two genes, or you work on the other one also?
So we work on Sentinel-2.
Yeah.
But I have the good news actually that the company that I co-founded named NeuroNOS was granted by the FDA to start clinical trials in PMS syndrome that has SHANK3 mutation, Phelan-McDermid syndrome. So we were granted by the FDA last week to do clinical trials. Of course, NeuroNOS now is doing toxicological studies, so we can start clinical trials in 2026.
So you are not working directly on autism yet, but you'll see the pharmacology and everything, the application for autism, because I remember when you were able to inhibit nitric oxide production in autism-like behavior in mice, you could completely reverse symptoms.
Right. So no, we work on autism actually, autism-like phenotypes in mouse models and in stem cells, and we look on core symptoms of behavioral autism such as social deficits, communication, anxiety, and also on neuronal and neurobiological mechanisms such as synaptic proteins, GABAergic and glutamatergic systems, how they are affected by nitric oxide in autism models.
I know you can already eat beetroot, but that's for endothelial. They say green.
Yeah, no. So we, as I said, we are developing small molecule to inhibit neuronal nitric oxide synthase. We co-founded a company that now has the molecule that we developed. The company now has already synthesized 1.5 kilograms to start toxicological studies in rats and dogs. So we have the molecule, we are in advanced stage in the preclinical studies, and we are aiming, as I say, to start clinical trials with this small molecule in 2026.
Fantastic. So your team was the first one to show an overproduction of NO in autism and Alzheimer?
Right, not Alzheimer's. We are the first to show a link between nitric oxide and autism and prove that overproduction of NO may lead to autism-like behavior and phenotypes.
But Alzheimer was discovered before.
Alzheimer's, Professor Stuart Lipton and others already showed this, many years ago.
And always it affects the synapse in both cases.
Yes, it affects the synapse and the neurons and the communication between them. In Alzheimer's, it leads to neuronal death. The NO overproduction may lead to neuronal death.
Where's the neuronal nitric oxide release? So directly from specific neuron? Do we know glia, astrocyte?
It's a great question. We know that most of the neuronal nitric oxide synthase is in the neuron, in the cells. It is located in the postsynapse. And since nitric oxide is a diffusible molecule, so we can find nitric oxide not only in the neurons, but also in microglia and astrocytes. So in microglia, there is also inducible nitric oxide synthase that is activated once the immune system is activated. And of course, we have the ability to identify which proteins are modified by nitric oxide, named S-nitrosylation.
So we know when we look at nitric oxide, remember, they describe it as a gas. Is it in form of gas from one neuron to the other neuron?
As you said, it's a gaseous and diffusible molecule, meaning that with a very short lifetime.
Right.
So meaning when NO is released, as I said, it's diffused and it can act in a different ways. Either nitrosylation, meaning modifying proteins and affect the protein's function, or leads to nitration of the proteins, which may lead to DNA damage and protein damage, or it can act as different other molecules such as nitrate, nitrite, and GSNO. So NO can activate other pathways in parallel and can affect different signaling pathways in parallel. So even if the NO has a short lifetime, it affects other signaling pathways that can stay for longer time.
Does he modify the immune system response?
There are many papers showing connection between nitric oxide and the immune system, and imbalance of NO can lead to autoimmune diseases such as Crohn's disease and colitis and etc. And of course, you as a doctor may know that we absolutely know that there is a link between NO and autoimmune diseases. By the way, we are also working on environmental factors. We just got a grant of $17 million from the California Institute for Regenerative Medicine, led by Professor Stuart Lipton, and my lab is part of this consortium. And we are trying to understand how air pollution affects autism and how nitric oxide in air pollution affects autism. So we are talking about nitric oxide first as an endogenous molecule, but second also can be as an exogenous molecule that can affect to brain damage.
Wow. So in air pollution, you have also an excess of NO, of nitric oxide?
Yes.
All right. So if you already have a tendency to have too much, then you have a little bit more from external source. And
yes, we are investigating this right now. We are now actually doing this work, exposing animal brains and human organoids brains to air pollution and testing the consequences of this on the brain and whether it leads to autism, Alzheimer's, or other neurological disorders.
So when we talk about the three isoforms of nitric oxide, so we have, again, neuronal, inducible, and endothelial. The molecule itself, is it the same exact molecule? It's just a different location?
You mean the structure of the enzyme?
Yeah,
so they are very similar.
They're not exactly the same.
Yeah, but they are not the same, absolutely. And this is why there is a lot of challenge in developing molecules that's specific for specific NOS.
So for example, the nitric oxide from the air pollution is more, you resemble more the neuronal, the endothelial. Which one it will?
Great question. Actually, the air pollution, what we know right now that NO in the air pollution and other molecules can induce the activity of inducible nitric oxide synthase, meaning that it can activate inflammation and a release of NO as a consequence of the activation of inducible nitric oxide synthase.
If I bring the same NO in the brain or in the vessel, they have the same action.
Yes, the NO itself, right.
Too much NO in the environment, you could have an endothelial action on you and dilate your vessel. Because if you your blood from
so we are the levels of NO in the air pollution we are talking about will not affect hypertension on vasodilation. It's not so high, that it goes into the vessels. We are talking about a good amount of NO that can get into the brain and make some toxic effect. And of course, it can also affect other organs, but we don't investigate other organs. But I'm pretty sure that the same molecules that in the air pollution will not affect only the brain, but also will affect other organs in a toxic effect.
So the NO is a small molecule, so I assume it crosses the blood-brain barrier, 20 nanometers.
Yes.
So once you produce a lot of NO, how does it affect the synapse in autism? What does it do exactly?
That's a great question. So what we found in our advanced science paper that published in 2023 is that when you give NO donor to wild type mice, you reduce the number of spines on the synapse. And also you reduce the levels of synaptic proteins. And when you give the NO inhibitor, you can see reversal of the regeneration of the spines in the neuron. So what we say is that NO is involved in the synaptogenesis and neurogenesis, meaning NO is involved in building up the neurons and the synapses, and therefore it plays a critical role in the brain.
Wow. And do we have a certain amount of lapse of time? I mean, you can have autistic child with chronic problem with their synapse, that have this problem for a longer time. Do we know if that's going to be reversible after a certain amount of chronic problem on the synapse?
Yeah, so we didn't check this, but we certainly know that the drug will be given in a chronic use. We don't think that our molecule will cure autism, but absolutely will treat the core symptoms of autism. That for now, there are no drugs for autism. Most of the drugs that are given are drugs that are given to schizophrenia and bipolar disorders, such as Risperdal and ariprazole. And these drugs that are given to kids have a great and severe side effects. So
Right. So we know that SHANK3, I think I remember 1 or 2% of the autistic case have a problem with SHANK3. How many percent of the autism you think will have some NO overproduction?
Yeah, this is a great question, actually. According to our analysis, we think that many kids on the spectrum has increase of nitric oxide. We are sure that low functioning of autism has increase of nitric oxide much more than the high functioning, but we still think that high functioning still can get the drug, but maybe in a lower doses. We're still not on that stage to decide who's gonna take the medicine. But I mean, we know absolutely that it's increased, at least in the low functioning autism, according to the blood testing that we did in the paper that was published 2023.
Most of the human low functioning autism may benefit from this drug.
Yes.
Perfect. And how do you test NO if you have a kid, the neuronal nitric oxide? How do you test it?
In the blood, we test nitric oxide biomarkers.
Right.
We don't test the enzyme. So we look for nitrotyrosine, we look for NO2, we look for NO3, we look on different biomarkers that mimic the amount of NO.
The whole NO, not specifically the brain or
Yes, absolutely.
Can you
Absolutely.
find those in the urines also?
So we didn't test it in urine, but I have to say that we got fibroblasts from autistic kids, and we differentiated into stem cells and into neurons, and we found increase of NO in these cells. This takes me to the second company that I co-founded, and we are now developing a blood testing for diagnosing of autism through multi-omics platform using AI models and algorithms in order to diagnose autism through simple blood testing.
So not just NO, but many other molecules.
We look for different proteins that are modified not only by nitric oxide, but also other different post-translational modification.
Perfect. But you think any day we could just do that in the urine, or they are?
Absolutely, there are some works that are trying to find in urine, but we personally didn't try it.
Okay, perfect. Now, synaptic pathology is closely related to mitochondrial dysfunction. Do you want to make the connection with the mitochondria? Because you also wrote an article on that.
Yeah, so since nitric oxide is linked to mitochondria, we are working now on how mitochondrial dysfunction leads to neurological disorders. One of the problems is the production of ATP and the leak of hydrogens when we are talking about ATP synthase. We identified in autism a hydrogen leak or H plus leak that will affect the production of ATP. And actually we did several tests on the mitochondrial function, starting from seahorse testing, from potential the membrane of the mitochondria, and other different parameters, and we see that there is a significant mitochondrial dysfunction in autism. And therefore, we think that if we block the leak in different molecules, we can reverse the mitochondrial dysfunction and the autism-like phenotype.
So the H plus leaking between the two membranes of the mitochondria or outside completely?
It is leaked outside of the mitochondria.
Outside.
And we think that there is a less production of ATP and then mitochondrial dysfunction.
And 93% of the ATP necessary for normal brain function is produced by the mitochondria.
Exactly.
So connection nitric oxide and mitochondria.
Yes, so of course, part of the production of nitric oxide also occurs in the mitochondria. So we think that we still don't understand the full mechanism of the crosstalk between the NO in the mitochondria and NO in the cytoplasm. It's a totally different study that needs to be done. But of course, we totally know that mitochondrial dysfunction leads to NO imbalance, meaning it affects the NO production and the balance of NO in the cell.
So when we say NO is produced in the neurons, do we know where in the neuron, how much in the mitochondria, how much in the?
Yeah, so this part is still under the investigated. We still don't know what is the ratio and whether it's important or not.
It would be like the endoplasmic reticulum, maybe, where NO could be produced.
Yeah, so what I'm saying is that we know that nitric oxide could be produced in the cytoplasm and in the mitochondria as well. We didn't test the cellular compartment aspect, I have to be honest, cellular compartment aspect of NO production. And this is a super important question that can be, answered. And I'm sure many other people worked on it trying to understand the location of the production.
Yeah, because the calcium, as you know, is connected with mitochondria, endoplasmic reticulum. That's why I mentioned it, extracellular space.
Of course. Several papers that I published at MIT showed that NO can nitrosylate endoplasmic reticulum channels in autism. And we think that nitrosylation of calcium channels also can affect the calcium influx, either by one or other direction.
Wow, that's really interesting. What about what we're thinking for the longest time, accumulation of toxins in autism?
Yeah. So this also started to investigate at MIT and then at the Hebrew University and now at Harvard. We investigated arsenic. It's a toxin metal that is found in water and rice and baby-based products. And we had two papers on arsenic. We exposed arsenic to animals and we found an intensive reprogramming of the biochemistry in the brain, and also it affects the behavioral of the mice in an autism and memory loss way. So we think that there are many toxins also in the food that may lead to dysfunctions.
So could you summarize? You have now two companies. So what do they work exactly on, and maybe any timeline when the public will be able to benefit from it?
Yeah, so the first company, as I mentioned, is NeuroNOS. It's a pharma company that develop small molecule and drugs for the treatment of autism, Alzheimer's, and brain cancers. And we are now in a intensive work to finish the IND enabling studies so we can start hopefully in 2026 or 2027 clinical trials. And the second company is Point 6 Bio. It's a diagnostic company that develop blood testing to diagnose autism.
So why Alzheimer? Because the connection is again with NO or more melatonin, or whether you...
Yeah, so absolutely. So nitric oxide is linked also to Alzheimer's. We published paper on Alzheimer's and nitric oxide on post-mortem tissues of humans, together with Professor Stuart Lipton from Scripps Institute and Professor Stephen Tenenbaum from MIT. We analyzed the brains of Alzheimer's, and we found that there is a unique modification of nitric oxide in women comparing to men. And we explained that this modification may be the reason for the increase of Alzheimer's cases in women comparing to men.
And also increase in production of nitric oxide.
Yes.
And what would happen in an adult? Again, it could be environmental, exogenous factors.
Yes, so in Alzheimer's, they are also genetic, but not as much as autism. There are many now studies. Some are saying it's a viral infection. Some are saying it's environmental factors. So a lot of research are being done there.
Fantastic. And you mentioned a 17 million grant. What kind of grant you have for your company?
We received many grants to fund our research. The first that I can mention is the Eagles Autism Foundation that is led by Mr. Jeff Flory in Philadelphia, which belongs to the Eagles Philadelphia team. I was the first non-American scientist to receive this prestigious grant. We also received a grant from the United States Department of Defense, from the German Scientific Administration. Recently, as I said, we received the $17 million, $17 million from the California Institute for Regenerative Medicine, led by Stuart Lipton, to investigate air pollution, nitric oxide, and autism.
How long before you think the public will be able to benefit from for autism or Alzheimer?
So, as you know, as the public knows, it is a long journey. I think we did a great job in proving the mechanism of action and proving the preclinical studies. And now the journey is long. It may take many years, but we hope at the end of the day that the drug that we are developing will benefit millions.
Right.
I have to say that I receive hundreds of emails from all over the world, from parents and families want to be updated on the development of the drug, and we work very hard in order to get this mission done.
Waiting. Wow, what an interesting discussion, Haitham. I know you're really, really busy. Thank you so much. Well, I'll keep in touch to see what happened with those discovery in a few years. Hopefully we'll have millions of people taking your product. If they are not toxic, you're on the toxicology level now.
Thank you so much. Thank you so much.
Pleasure.